Welcome to Biotanemy.
Biological impact of polychlorinated biphenyls and dioxins. Polychlorinated biphenyls or PCBs were first synthesized in 1881 by the German chemist Schmidt. However, large-scale industrial production began in 1929 by Monsanto in the United States under the trade name Arichlor. The PCBs consist of two benzene rings, 1 to 10 chlorine atoms substituted at various positions, and 209 possible congeners. They were developed because of their chemical stability, non-flammability, high dielectric constant, thermal resistance, and low electrical conductivity. The properties that made them ideal for industrial applications during rapid electrification in the 20th century. They are used in transformer and capacitor coolants, hydraulic fluids, plasticizers in paints and sealants, carbonless copy paper, flame retardants, and lubricants in turbines. Their chemical inertness and thermal stability were considered technological advantages at the time. The toxic trail of PCBs. PCBs are produced by the chlorination of biphenyl. During this process, byproducts like chlorinated dibenzofurans, dioxin-like compounds such as 2,3,7,8-tetrachlorodibenzopdioxin, and hydrochloric acid vapors, are produced. During their operational lifetime, PCBs were released into the environment through multiple pathways, including leakage from transformers and capacitors, volatilization from PCB-containing paints and sealants, emission into indoor air, particularly in older buildings, and accidental fires, which not only dispersed PCBs but also generated highly toxic byproducts such as dioxins and furans. Also, the improper incineration produces polychlorinated dibenzodioxins, polychlorinated dibenzofurans, and highly toxic combustion byproducts. The landfill leakage contaminates soil, groundwater, and rivers and marine ecosystems. PCBs resist chemical degradation, they are not easily hydrolyzed, have half-lives of decades and undergo long-range atmospheric transport. Due to global concerns, PCBs were banned under the United States Environmental Protection Agency in 1979 and later globally restricted under the Stockholm Convention on Persistent Organic Pollutants. Biological effects and side effects. PCBs possess several structural characteristics that account for their strong lipophilicity and environmental persistence. Their core structure consists of two benzene rings forming an extensive aromatic system, which creates a hydrophobic pi-electron cloud that disfavors interaction with water. The presence of multiple carbon-chlorine bonds further enhances their nonpolar character, as these bonds are relatively unreactive and poorly soluble in aqueous environments. Increasing chlorine substitution not only raises the molecular weight but also significantly increases hydrophobicity while eliminating polarity, since PCBs lack ionizable functional groups. This pronounced lipid affinity is quantitatively reflected by their high octanol water partition coefficient, which indicates a strong tendency to partition into biological membranes and fatty tissues rather than aqueous compartments. After ingestion or inhalation, they circulate bound to lipoproteins. They diffuse passively across cell membranes, which are lipid bilayers, and accumulate in adipocytes, where neutral triglycerides provide a hydrophobic storage matrix. Adipose tissue acts as a long-term reservoir and slow-release source into the bloodstream, which explains why chronic internal exposure can persist even after external contact has ceased. Furthermore, physiological conditions such as weight loss or pregnancy can mobilize stored lipids, leading to the release of accumulated PCBs back into circulation and increasing systemic exposure. Because PCBs are small, nonpolar, and highly lipid-soluble molecules, they readily cross the placental barrier through passive diffusion and accumulate in breast milk due to its high fat content. As a result, maternal exposure can lead to fetal neurodevelopmental toxicity during pregnancy and immune as well as endocrine disruption in newborns during lactation. Certain coplanar PCB congeners structurally resemble dioxins because their planar configuration allows them to fit into the ligand-binding pocket of the aryl hydrocarbon receptor, AHR. The most extensively studied prototype ligand for AHR is 2,3,7,8-tetrachlorodibenzopdioxin, which serves as a model for understanding dioxin-like PCB activity. PCBs, particularly the coplanar or dioxin-like congeners, exert toxicity primarily by activating the aryl hydrocarbon receptor or AHR.
After diffusing into the cytoplasm, PCBs bind to AHR,
which normally exists in an inactive complex with chaperone proteins such as HSP90. Ligand binding induces a conformational change that enables the PCB-AHR complex to translocate into the nucleus, where it dimerizes with ARNT or AHR nuclear translocator. This heterodimer binds to xenobiotic response elements in DNA, initiating transcription of genes involved in xenobiotic metabolism, cell cycle regulation, apoptosis, and inflammatory signaling. Although this pathway physiologically contributes to detoxification, persistent stimulation by highly stable PCBs leads to sustained gene dysregulation. Consequently, cytochrome P450 mediated metabolism is often incomplete, generating reactive intermediates such as epoxides and quinone derivatives. These electrophilic metabolites can form covalent adducts with DNA and proteins, initiate lipid peroxidation, and disrupt membrane integrity, thereby promoting mutagenesis and cellular dysfunction. A major downstream consequence of these processes is oxidative stress. Reactive oxygen species arise through redox cycling of hydroxylated PCB metabolites and increased electron leakage associated with enhanced CYP activity. Additionally, PCBs impair mitochondrial function by disrupting membrane structure and interfering with oxidative phosphorylation, further increasing electron escape and ROS formation. Excess ROS damages lipids, nucleic acids, and proteins, activates redox-sensitive transcription factors such as NF-kappa B, and alters cellular fate decisions, leading either to apoptosis or aberrant proliferation. These damages will lead to carcinogenicity, endocrine disruption, neurotoxicity, immunotoxicity, reproductive toxicity, and dermatological effects. Solutions and remediation strategies. Because PCBs are highly persistent and resistant to natural degradation, their elimination is technically complex and requires integrated preventive, disposal, and remediation approaches. Prevention remains the first line of control and includes strict international bans, safe storage and phased removal of old transformers and capacitors, and systematic identification and labeling of PCB-containing materials to prevent accidental release. For safe disposal, high-temperature incineration at temperatures exceeding approximately 1,200 degrees Celsius is widely used to achieve complete thermal destruction, although strict operational control is necessary to prevent the secondary formation of dioxins and furans. Plasma arc destruction, which operates at extremely high temperatures, provides effective molecular breakdown of PCB compounds. Chemical dechlorination methods, including base-catalyzed decomposition, reduce toxicity by removing chlorine atoms from the biphenyl structure, thereby decreasing persistence and bioaccumulation potential. Environmental cleanup strategies such as bioremediation and phytoremediation focus on contaminated soils and sediments. Bioremediation employs specific microorganisms, such as species of the genus Burkholderia, capable of degrading lower chlorinated PCB congeners through aerobic and anaerobic reductive dechlorination pathways. Phytoremediation enhances microbial degradation within the rhizosphere by utilizing plant-microbe interactions. In heavily contaminated aquatic systems, sediment dredging has been implemented, as demonstrated by remediation efforts in the Hudson River following industrial discharge. Additionally, activated carbon treatment is used to adsorb PCBs from water bodies, thereby reducing bioavailability and ecological exposure. PCBs remain a significant environmental and public health concern because they persist in soils and sediments for decades, bioaccumulate in aquatic organisms such as fish, and undergo global atmospheric transport that distributes them far beyond their original sources. In addition to their environmental persistence, emerging evidence suggests transgenerational epigenetic effects, indicating that exposure may influence health outcomes across multiple generations. Consequently, even decades after regulatory bans, PCBs continue to be detectable in human blood samples worldwide, reflecting their long-term stability and ongoing exposure through the food chain.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.